At the most fundamental level of human existence, there is a process happening inside your body that determines whether life itself continues beyond a single generation. It is not visible. It is not conscious. And yet, it is one of the most precisely regulated biological systems ever discovered. Because inside the male reproductive system, trillions of molecular decisions are being made every day to ensure that a single cell, among millions, carries forward the complete blueprint of human life. This is not just anatomy, it is information biology, hormonal control, cellular division, genetic recombination, and evolutionary continuity, all operating as a single integrated system. And it all begins in the brain. Deep within the hypothalamus, a small cluster of neurons acts as the master regulator of reproduction. These neurons release a hormone called GnRH in a pulsatile rhythm. Not continuously, not randomly, but in carefully timed pulses that determine the entire downstream cascade of male reproductive function. Those pulses travel to the pituitary gland, a small but powerful endocrine structure located at the base of the brain. In response, the anterior pituitary releases two critical hormones into the bloodstream, LH, or luteinizing hormone, and FSH, follicle-stimulating hormone. Together, they form the central signaling system of the hypothalamic pituitary-gonadal axis, a feedback loop that connects the brain directly to the testes. LH travels through the bloodstream until it reaches specialized cells in the testes called Leydig cells. Here, it triggers the production of testosterone, a steroid hormone that does far more than influence secondary sexual characteristics. Testosterone regulates gene expression, supports spermatogenesis, modulates metabolism, and feeds back into the brain to regulate the entire system. FSH, on the other hand, targets Sertoli cells. Located inside the seminiferous tubules, these cells are essential for nurturing developing germ cells. They provide structural support, nutritional factors, and biochemical signals required for sperm development. But to understand the true complexity of this system, we must go deeper. Inside the testes, a process unfolds continuously. Spermatogenesis, a highly organized sequence of cellular differentiation that transforms undifferentiated germ cells into highly specialized spermatozoa. This process begins with spermatogonia, stem cell-like precursors that divide through mitosis to maintain a constant population while producing cells destined for meiosis. Then comes one of the most important transitions in biology, meiosis, a specialized form of cell division that reduces the chromosome number by half. This is essential, because when sperm and egg unite, the full genetic complement must be restored. During meiosis, DNA is not simply copied, it is reshuffled. Paired chromosomes exchange genetic material in a process called recombination. This generates genetic diversity, ensuring that no two sperm cells are genetically identical, the biological engine of variation itself. Genui, biology_learning_block. Type _I_D, meiosis. From a single germ cell, 4 haploid sperm cells are eventually produced, each one carrying a unique combination of genetic information. But producing genetic material is not enough. Sperm must also undergo maturation. Inside the seminiferous tubules, Sertoli cells guide developing sperm through tightly regulated stages. They control the microenvironment, maintain the blood-testis barrier, and ensure that only properly developed cells proceed. Cells that fail quality control are eliminated, recycled, reabsorbed. Nothing is wasted. Once maturation is complete, sperm cells are released into the lumen of the tubules and transported to the epididymis. Here, they undergo final functional maturation, acquiring motility, membrane stability, and the ability to recognize and penetrate an egg. But even at this stage, they are not yet active participants in reproduction. They remain stored, quiescent, waiting. Because without proper hormonal stimulation, the system remains partially suppressed. This is where endocrine regulation becomes critical. The hypothalamic pituitary gonadal axis operates as a closed feedback loop. Testosterone produced in the testes travels back to the hypothalamus and pituitary. When levels are sufficient, GnRH and LH secretion decrease. When levels fall, secretion increases. This dynamic feedback ensures balance, preventing both underproduction and overproduction of reproductive hormones. But testosterone is not the only regulatory molecule involved. Inhibin, produced by Sertoli cells, provides additional feedback specifically targeting FSH secretion. This allows extremely fine control over spermatogenesis, adjusting sperm production in response to physiological conditions. Stress, nutrition, temperature, and overall metabolic state all influence this axis, because reproduction is biologically expensive, and the body only invests in it when conditions are favorable. At the cellular level, sperm development is one of the most energy-demanding processes in human biology. Mitochondria play a crucial role here, especially in developing sperm cells where energy production must be tightly coordinated with structural changes. Chromatin undergoes extreme condensation. Cytoplasm is reduced. A flagellum forms, creating the motile structure necessary for movement. Each sperm cell becomes a highly specialized vehicle designed for a single purpose: to deliver genetic material, nothing more, nothing less. And yet, despite this apparent simplicity, the system that produces sperm is extraordinarily sensitive to disruption. Elevated temperature can impair development. Hormonal imbalances can reduce production. Genetic mutations can halt the process entirely. Even environmental factors such as endocrine-disrupting chemicals can interfere with signaling pathways. This fragility highlights a deeper truth. The continuity of human life depends on a system that is both robust and delicate at the same time, capable of producing millions of cells daily, yet vulnerable to subtle changes in internal chemistry. As sperm travel through the reproductive tract, they encounter additional layers of regulation. Epididymal secretions modify membrane proteins. Seminal fluid from accessory glands provides nutrients and buffering capacity. The prostate contributes enzymes that activate sperm motility after ejaculation. Each step is precisely coordinated, not random, not redundant, but evolutionarily optimized for efficiency and survival. At the moment of ejaculation, millions of sperm cells are released. Yet only a fraction will survive the journey through the female reproductive tract. An even smaller fraction will reach the egg, and only one will succeed in fertilization. A biological bottleneck of extreme selectivity, where competition is not just between organisms, but between cells carrying slightly different versions of the same genetic blueprint. And through this process, genetic continuity is maintained, generation after generation, not through intention, not through awareness, but through molecular systems operating with extraordinary precision. The male reproductive system is often described in simple mechanical terms, but in reality it is a deeply integrated endocrine and genetic control network. A system where the brain, glands, and gonads function as one coordinated axis, where hormones act as information signals, and where cell division becomes a mechanism for biological diversity. And perhaps the most remarkable aspect of all is this: at no point does the system decide in a conscious sense. There is no awareness, only regulation, feedback, chemistry, and time. Yet from this molecular choreography emerges one of the most fundamental outcomes in biology: the continuation of human life itself.